Hello, everyone. It's a pleasure to be here. So I'm very excited. Of talking about our progress in the multifluorescent mini brain organoid to test neurotoxicity, to do neurotoxicity screening. So in first place, I would like to acknowledge all my awesome team and collaborators, as well as our funding that was mainly from EPA. As a background, I would like to dive in straight to the problem that we are trying to tackle. So we have a really limited knowledge about the neurotoxicity properties of the chemicals that we are surrounded with throughout our life, more so during development. So to illustrate this, you can see here that we have like more than 100,000 chemicals in the world. From this, we only have identified 1,000 as neurotoxic, and from those, we know that around 200 are neurotoxic for humans, and only 14 we have identified as toxic to neurodevelopment. Even more striking is being published that gestational exposure to environmental chemicals has a transgenerational impact, what makes all this more relevant. In response to this, US EPA and OECD, they created new guidelines for the developmental neurotoxicology. However, these guidelines are they are based heavily in animal testing, which we all know that they have major limitations. For example, 90% of clinical trials fail, and then that is related to the intraspecific differences. Also, they are very low throughput, very expensive, and time and labor consuming. And last, it brings up several ethical concerns. In response to this, new approach methodologies emerged. So then there are new alternatives to test chemical toxicity by using in vitro, in silico, or omics-based approaches. So here I'm showing in a very simple schema the key events of neural development at the cellular level. So we have like 2D systems to assess some of these different key events to assess the chemical toxicity. So, but then they are 2D. We screen them independently. They are independently, and they are targeted for the specific cell type that we want to work. Also we are missing other key events as myelination in these guidelines. So now we have so many technology like 3D macrophysiological systems. So we wanted to, in effort to that, in our lab, we developed this brain sphere model from iPSCs. This model has different types of neurons, astrocytes, oligodendrocytes, and is electrophysiologically active. This way, the goal of my project was to generate a 6-in-1 brain sphere assay to use as a relevant alternative to animal testing, where most of the key events for neurodevelopment can be covered in one 3D brain system and assessed simultaneously from one sample. In order to do this, then we selected like specific markers for each of the cell types that we have in our brain spheres. For example, if we target PLP, we will be able to assay myelination. If we target GFAP, glia migration and gliosis. Same with neurons, we will be able to see neurodifferentiation and neurite growth. And last, if we tag synaptophysin, then we'll be able to follow synaptogenesis. And in combination, we can describe our neural network. Also, as part of this project, we have developed a 3D microelectrode array. So if you want more details about it, you can visit the poster 234 on Thursday, when she will be talking about it. Okay, so very briefly, we use a CRISPR-Cas9 technology to insert these tags of interest. So we design the plasmids and we deliver them using lipofectamine. Then we provided a template for the cells to be able to repair the double-strand break caused by Cas9 and incorporate the fluorescent tag in the process. After this, we enrich the population of genetically modified cells using pyromycin, and then we did the PCR screening to test the efficiency of the transfection, as well as the limiting dilution to get isogenic clones. Okay, so we did 2 different methods of transfection, reverse and forward transfection. The most efficient one was reverse transfection, that is when you add the complexes and the cells at the same time. So here I'm showing like the primers that we use to design to screen the efficiency of the tag. So here in the orange, you are seeing the upper band is the knock-in incorporation and the lower band is the wild type. This is going to show us like the efficiency that we have compared to the wild type, the cells that still have wild type. And then the second part, it will show us the actual incorporation in the genome, because the primers are attaching to the area that is not inside the plasmid. Then this is a representative agarose gel showing the different clones that we screen. So this is very nice because we improve the efficiency compared to other methods where you usually have to screen 100 clones to be able to get some positive clones. Here we found that we increased the heterozygous incorporation, but even more so was the homozygous insertion. So we had like about 10 clones that we are ready to screen that are fully knock-in. So then, after selecting the clones, then we proceeded with the pipeline to screen all these that they have everything correct. So we sequence here all the area that was inserted, as well as the part of the gene of interest. Then we use a stem cell technologies kit to screen for the most common mutation sites in iPSCs. So here you can see we found 2 clones that they had some putative mutations, so we had to discard those. And then we screened the off-target effects, we sequenced them. In the case of PLP, we found that we hit one of the off-target effects with a 25 to 40% penetrance. And then we did STRF profiling to authenticate the cell line. And then we did flow to just check the pluripotency of the clones, as well as the neural induction potential. Then after this, it was very important for us, because you can go through all this process and then at the end the tag doesn't light up or impairs the function of your protein. So this is very important, and it's very hard to do in iPSCs. So we observed that actually our tag light up. We can see the GFP here, and then we did gene expression to look at the expression of PLP and GFP, and we saw a strong positive correlation. We also did a flow cytometry, so you can see here the expression of GFP, as well as the colocalization when you stain the cells with PLP antibody. We wanted to further characterize that the tag was only expressed, it was specific for oligodendrocytes. So then we stained with myelin basic protein, that is another oligodendrocyte marker. So here you can see the endogenous. Well, because of the light, you cannot see that much, but you can see here the whole sphere. You see some colocalization. Here, zoom in, you can see a nice oligodendrocyte, and you can see the colocalization of the endogenous and the pseudo-staining. And here in a zoom in, hopefully, you can see, I'm trying to point out here that you can find 2 different morphologies, and this is something that we can use to screen different toxicants. We have different morphology. This is oligodendrocytes when they are with MVP and the endogenous. So you can see processes compared to the, for example, the progenitor cells. Oh, this was what they should point out only to the ones that have green. Okay, so these that they have few or no processes. So these are OPCs. This way we can quantify oligodendrocyte progenitors, oligodendrocytes. Also, we can, real time, we can screen the differentiation of the OPCs as well as the oligos maturation. Then we stain our brain spheres with neurofilament heavy chain to show the close interaction and the functionality of the protein after the genetic screening. And we can see the close interaction here with the axons and the endogenous protein, and then this is a Z-stack, and doing a 3D reconstruction, you can see the nice wrapping of PLP around the axons, suggesting early myelin. This is at 8 weeks. Then when we move to 10 weeks, we use tubulin 3 as a marker of neurons. Again, we use MVP it's a mature oligodendrocyte marker, and then you can see the nice colocalization, and we can see how this network grew and it's richer. If you zoom out to some of these nodes that you see the colocalization of both proteins and then very close to the axons, you can see here with a 3D reconstruction, hopefully, okay, cool. So then you can see the nice wrapping of these two proteins around the axons, showing that we have myelin in our model. So this is very important because it's, this is one of the key events that we haven't addressed in DNT testing. Okay, so to validate our talk, then we challenge our brain spheres with cupresson. Cupresson is a demyelinating agent that is being used in murine models to study multiple sclerosis. The mechanism is not totally understood, but we know that it induces apoptosis of mature oligodendrocytes and subsequent demyelination. Contrary to what we expected, we saw a significant increase in the expression of PLP and therefore GFP. Then we checked the gene expression of both MVP and PLP, and we didn't observe any differences. But after looking at the literature, we found a paper showing that mouse cortex, that they saw increased proliferation of OPCs in the mouse cortex after cupresson expression. So we then checked the gene expression of NG2, that is one well-known OPC marker, and we saw a significant increase. Then we applied the technique that I showed you before when we quantified the OPCs, only the green cells, not the MVP positive cells, and we observed that there was also an increase. So we can say that we observed the same phenomena in our brain spheres as a compensatory mechanism. Okay, so after this took like over a year to get the first tag, to do all the, this is very hard, like it's not the same as doing knockouts. So knock-ins are very challenging. So then we chose the clone that performed best, and we started doing this second tag that was synaptophysin with blue. So here, please bear with me because the light won't help for you to see the blue. And so then, again, this is the efficiency of the transfection. That is very nice, the knock-in, and then the incorporation in the genome. So here you can see at 8 weeks, both endogenous, the oligodendrocytes, and then the dots of the punti of the synaptophysin. Here is 10 weeks, so you can see, and this is the pseudostaining with synapsin. So this is very hard to see, so I'm pointing out the dots. The next one is a higher magnification, so yeah, I think you can see better here the blue dots with this is PSD95, that is a postsynaptic marker. So in summary, I'm very happy with the progress so far in the middle of pandemic and everything. So far we've had 2 tags. We are halfway, so we are actively working in the other 2 tags. In the meantime, also collaborating with Dr. Berlinike, she's doing the high content imaging. So every time that I get a new tag, I will send her a bunch of brain spheres, and she will create algorithms to scan these mini brains, doing C-stacks with confocal, and then she will create the masks, and then we can determine the parameters that we are going to use to do the chemical testing. In the same way, we are going to do chemical exposure to non-neurotoxicants. That way we can establish functional toxicity endpoints based in the high content imaging. And then we will define testing strategies for DNT and write the SOPs. And we really hope to reduce the cost and accelerate prioritization and testing of environmental chemicals, as well as reduce the use of animals in neurotoxicity studies. Thank you so much for listening, and I'm happy to answer questions.